If you want to study a construct with mutations introduced by site-directed mutagenesis or a variant of your gene, begin by determining which sequence is the longest version of your gene. You will need to order this sequence using the GeneArt Gene Synthesis service and set up the shorter version as a variant. Let's go to the GeneArt customer portal and get started. To set up the synthesis of the longest version of your gene, drag and drop the gene synthesis icon as shown. Read the biosafety confirmation. Select the biosafety requirements for your project and click Save. Next, work through the gene synthesis form by double-clicking the gene synthesis icon and entering the requested data and information. Note that if you choose to optimize your sequence using the GeneArt GeneOptimizer service, all variant sequences must be based on the optimized sequence. When you have completed the gene synthesis form, the status indicator should read Complete. To learn more about completing the gene synthesis form, please watch the gene synthesis tutorial. Now let's see the process for ordering a single variant using the GeneArt portal. To set up your variant, drag and drop the single variant icon onto the gene synthesis icon as shown. The red exclamation mark seen here indicates that more information is needed. The next step is to open the single variant form by double-clicking on the single variant icon. Enter a name for your sequence. In this example, we've chosen the name EPO variant. The next step is to enter either a DNA or a protein sequence. Note that if you chose to optimize the sequence of the longest version of your gene when setting up the initial gene synthesis, the variant must be based on the optimized sequence. You can access the optimized sequence in the gene synthesis editor in the analysis report. Let's have a look at it. Just click on this button and download or view the optimized sequence. Now we'll return to the variant form. If you choose to enter the protein sequence rather than the DNA sequence, your project will require review by a GeneArt scientist. You will receive an email when the review is complete, and then you can continue the ordering process. In this example, we chose DNA. Copy and paste your variant sequence into the sequence field. If you plan to subclone your variant, choose the restriction sites you need in your sequence here. Note that the restriction sites must be present in the sequence you have entered. If you need extra services, select them here, then click Save. The status indicator should now read Complete. Place your order by clicking the Proceed to Summary button. Then you can add the project to your cart and proceed to checkout. Please view the Add to Cart tutorial to see how that process works. We have now used the single variant form for creation of a single variant. If you need several variants for your sequence, learn how to order them at the same time by viewing our Variant Bundles tutorial.